Okay, how are you? So this is my second presentation today. So for those that haven't heard it, at 10:30, you get to hear the full presentation at least. Earlier it was an interview, so now I get to show you some of the data that we and others collected, and so I promised earlier that I will show this slide, which is these are the major risk factors for so smoking and obesity, especially, are major risk factor for cancer, heart disease, Alzheimer's. And so I think that, of course, we need to reduce obesity and reduce smoking. But when you compare these risk factors with 30 years of aging, you see the impact of even smoking and obesity is very small and almost gone if you think about Alzheimer's disease compared to 30 years of aging, right? So meaning that aging is by far the most powerful enemy when it comes to mortality, when it comes to diseases. And so because it's so important, because there's a program that basically keeps us young and functional, we've been focusing not so much on longevity, but on what I call juventology and youth span, right? So can we study youth? How does a system, and this is true for all organisms, sorry, how does a simple organism stay young? And how do humans stay young for, let's say, 40, 45 years? And can we make that 45 years, can we extend that to 65 years, 70 years, right? So that's the idea of youth span and juventology. And then, of course, the rest of it is health span, right? Okay, now we're not young anymore. Can we stay at least healthy? And so if you look at, a lot of people, and journalists sometimes do this, and they love to do that. Say, oh, but Madame Calmont, she lived to 122 and smoked a pack of cigarettes a week. And she was proud of it, by the way, right? And so, yeah, then these lifestyle things don't mean anything. They're not that important. But it turns out, if you look at Chicago, for example, I lived a few years in Chicago, and there's a 30 years life expectancy difference between two zip codes, right? So depending on which zip code you belong to, you can have a 30-year difference in life expectancy. Now, of course, it's not just nutrition, it's not just fasting or running. There's a lot of other factors, socioeconomic, et cetera, but let's say that probably about 20 of those 30 years can be attributed to lifestyle. And that's what a lot of research suggests. So how do we, what controls the, so for thousands of years, people have been fascinated with aging and longevity and immortality. And so, and I think a lot of people, until the genetics of aging was established, meaning identification of genes that can actually make organisms live longer, healthier, right? Until that, which happened, and we were part of it in the 90s, people just thought, there's not much you can do about aging. That's the way it is. You're going to age. So we were working, if you look at the bottom, we were working, I was working with people, and then I said, this is too difficult to figure out genes that control aging in people. So I moved back to simple organism, like a unicellular organism. So one cell is an organism. And then we identified the genes that control aging in that organism. And it turns out that the genes that control aging were in the sugar pathway and in the protein pathway, right? So if you blocked the pathways, the set of genes that are activated when you have sugar in your system or you have proteins in your system, that organism lives 5 times longer. And if you then fasted that organism, now live 10 times longer, right? So a combination of fasting and genetic mutation made this revolutionize the lifespan of a simple organism. But it turns out that others have shown similar effects. In this mutation, at least the mutation in the protein pathway, cause dwarfism. You see, this is small, it's a colony of organisms compared to the normal one. And same thing here. This is the long-lived one, this is the normal one. Long-lived one, normal one. These little mice, they're lacking the growth hormone receptor. They're either the growth hormone or growth hormone receptor deficient. And now keep in mind that people take growth hormone to attempt to become younger, but it's exactly the opposite, right? So if you look at the data, it suggests that lots of growth hormone activity is accelerating the aging process. But the important thing about the little mice here that are lacking, don't have the growth hormone receptor. So it's almost like they don't make growth hormone. So the major hormone in the human body that makes you grow, but not just makes you grow, but makes a lot of cells grow. So these mice live 40% longer, but half of these mice never develop any disease. That's really remarkable, right? So imagine that, in the US, if we went from an average lifespan of about 80 years to an average lifespan of 120 years, and half of people would never get a disease of any kind, right? So I think that going back to the slide that I showed you earlier, this is possible. And so eventually we started a collaboration 20 years ago with Jaime Guevara in Ecuador, an endocrinologist in Quito, Ecuador. And Jaime, so we started looking at people that had the same mutation as these super long-lived mice. And it turns out that, I already showed you this. So they had a terrible lifestyle, right? So they drank, they smoked, they didn't exercise, had a terrible diet. And so when I first went down there, I thought, this is never going to work. I mean, this is just too much, right? The genes are not going to be that powerful. They're not going to be able to counterbalance this bad lifestyle. But sure enough, they did, right? So if you look at their relatives, they not die of cancer, about like it happens in the US, right? So about over 20% of the deaths were caused by cancer. But if you look at these little people, after 50 years of observation, almost nobody died of cancer. Very few people died of cancer. And a lot of people died, but they did not die of cancer. And if you look at diabetes, obesity, these little people actually have a higher level of obesity. So you would expect a very high level of diabetes. And instead, they rarely develop diabetes. And another thing, we eventually brought them to Los Angeles. And we brought them to USC and did what we call functional MRIs, right? So functional MRIs, we show them objects, and then we ask them, what did you just see? Do you see now a new object that is different, similar, or the same as you already seen, right? So it's a cognitive test. And when we did this cognitive test, these are the little people, and these are their relatives, same age, and they're related to each other. But if you look at the profile and don't worry about the details, but just look at the overall profile. You see, it's very similar to that of 20-year-olds controls, right? And if you look at the one from their relatives, it's very similar or more similar to the one of the 70-year-olds, right? So this is suggesting that just like for mice, cancer, diabetes, cognitive decline, just a single mutation is able to make these really remarkable effects. And again, because we see it both in mice and in people, it's unlikely that this is just a coincidence, right? And research, as I showed you earlier, even comes from fruit flies and yeast. So it's a very conserved set of observations. Okay, so this is just to show you, don't worry about the details, but to show you proteins and sugars are driving. This is the growth hormone that these little people are lacking. And if you want to increase that, you have a high protein diet, right? And if you want to increase this pathway here, then you have a high sugar diet. Okay, so what happens if you just decrease everything by 25%? And this is something that was done by Richard Weinrock that came out of the same lab as I was in at UCLA back in the 80s. He was in the Walford lab. And he started like a crazy experiment. He took a lot of monkeys, and then he exposed these monkeys to the same diet, same identical diet, but one had normal level of calories, and one had 25% reduce calories, right? And the results, I think, are remarkable. The monkeys that were on the calorie restriction diet, the normal monkeys had 60% of them had either diabetes or insulin resistance, or pre-diabetic. They went down to 5% in the calorie-restricted monkeys. Tumors in the calorie-restricted monkeys were reduced by 50%, and cardiovascular disease reduced by 20 to 30%. It's really such a simple intervention. But that, if you look at the deaths caused by these type of diseases, there's a big difference. These are the calorie-restricted monkeys. This is a survival, right? So about 60% are alive at 35 years of age or so, versus 25% in the control, right? So big difference. But if you look at all-cause mortality, so all reasons, all causes of death, then they more similar, right? So this is suggesting that, yes, the calorie restriction revolutionizes the risk of the major disease that kill people, but probably cause other problems, right? And there's a trade-off, right? So you don't get diabetes, you don't get cancer, but maybe some of these monkeys died during anesthesia, right? So they got anesthesia, they never woke up from anesthesia. And so the point is, how do we, and actually Walford is the bald guy there, and he was my first mentor during the PhD. And in 1992, when I joined the lab, he was not at UCLA, he was in Arizona, locked in this biosphere 2, sealed environment. And Walford, in the sealed environment, started the first human study on calorie restriction. So what I just told you for monkeys, he actually did it before the monkeys. He did it on people, a small group of people. It was only 8 of them, and they were inside of this biosphere. And he sort of found an excuse to make everybody calorie restricted, because it was not supposed to happen like this. But the food that they were growing inside was scarce, and he said, well, why don't we calorie restrict? So we save food. And not surprisingly, the effects were similar to what I just showed you for monkeys. Reduced markers for cancer, heart disease, diabetes, hypertension. And so these are just some examples. Systolic blood pressure, they were a healthy group, right? But they started 110 and they go to 90. Glucose, they started fasting glucose at 90, they go down to 70. Cholesterol, a normal 185 and they go down to 125 total cholesterol, right? So the extreme effects. And now you could argue that these are good effects, or you could argue these are bad effects, because maybe Walford was being pushed, as I mentioned for the monkeys, he was pushed over the limit, right? So Walford, this is the Walford when he came out of Biosphere 2. And I was actually there in Arizona when they came out. And they looked, very stressed out and very bad, right? So then there was my motivation 30 years ago to say, over 30 years ago, to say, this is not, I mean, obviously the effects on diabetes, cancer, cardiovascular disease are remarkable, but this is not the way to do it. And so how do we do it? And so we started thinking about, some of the species that fast for a long time. And for example, the emperor penguins of the South Pole, they fast for months every year. And, but they overeat for a period of the year so that they can fast for several months a year, incubating the eggs for the containing the offspring. And so I started thinking, what if we just intervene with a few days of fasting periodically, once in a while, let's say once a month or once every 3 or 4 months, and you just do 5 days of fasting. And that's what we did for cancer patients at USC back, nearly 20 years ago. So we thought we were seeing very strong results on fasting and protection of mice against chemotherapy. So we said, let's try it on people, and realized that nobody wanted to fast, right? Nobody wanted to do water-only fasting. The oncologists were worried, and the patients were worried about water-only fasting. And so we developed, thanks to NIH funding, we developed a fasting-mimicking diet. So it's a low-calorie, low-sugar, low-protein, high-fat, plant-based diet. So the idea is like, let the patient eat and get the effects of fasting still. And we knew this from lots of studies in mice, and we knew how, why to do this. Okay, so then one of the studies that I always wanted to do, and finally I got to do it very recently, was, so for 50 years, right, Ansel Keys defined the Mediterranean diet in southern Italy, came from Minnesota, and defined the Mediterranean diet 60 years ago or so, right? And after 60 years, even Italians are not on a Mediterranean diet anymore, right? I mean, I think less than 10% of Italians are doing the Mediterranean diet. So you could keep trying to convince the world that they should change the diet, right? Or you could say, okay, yeah, we try, we keep trying, and you should eat better and healthier, et cetera, et cetera. And, but let's come up with alternatives, right? So what if you take mice and you give them a terrible diet, a Western diet, like hamburger, cheese, and fries type of diet, and you do the same, but once a month you give them 5 days of this fasting mimicking diet, very similar to what I described for humans, but made for mice, right? So what's the effect of those 5 days? Even though the other 25 days a month you eat this bad diet, high sugar, high fat, high calorie. So what happens is this. Now, surprisingly, these are the mice on the high fat Western diet. These are the control mice. And look at what happens just 5 days a month of the FMD, right? Just 5 days a month. So now it becomes more reasonable, right? Somebody looks at it and says, I could probably do 5 days, right? Especially if you don't make it once a month. Can you make it every 3 or 4 months, right? So we don't know yet, but we'll see. But I'll show you clinical data in a second. But the other interesting thing is no loss, lots of fat lost, no lean body mass loss, right? So muscle and bone maintained intact, right? So these mice are doing very well, and even look at cholesterol, right? Cholesterol goes sky high in the older mice with this bad diet, and the FMD cycles are able to bring it back. So it's bringing everything back to normal, in spite of this terrible diet most of the time. Okay, so then the ultimate test is longevity. How long do they live? And this, I mean, we were really surprised, right? How bad the Western diet is in making, this is the normal diet, this is the Western diet, almost cutting the lifespan in half, right? So it's really, and this is going back to the Chicago, what I was saying about Chicago and the different neighborhoods, right? This is how bad, having a terrible diet, bad of an effect can have on your life. But, the just 5 days a month, the FMD didn't completely reverse these effects, but certainly most of it, right? Most of it is back to normal, even if we consider lifespan. Okay, so what about people? So we developed the human version of it, and then we ran a lot of trials. Now I'll show you a few examples, but there's now maybe 35 different clinical trials that have been run by universities all over the world, testing these many different diseases. But I'll show you a few examples. And so this is 1,100 calories on day 1, then it becomes 800 calories on day 2, 3, 4, 5, and that's a lot of vegetables, nuts, and a lot of very healthy. So it's combining the fasting effects with the blue zone type of food, right? So very healthy plus fasting mimicking, right? Fasting mimicking, right? So this is what we gave to patients. And so you see now very similar to the animals, body weight drops, abdominal fat drops, waist circumference drops, but lean body mass is not a factor, right? So they're maintaining their lean body mass, and then I'll show you in a second probably why that is. And if you look at, compared to the calorie restriction, remember the people, they went into the biosphere, they were healthy, but everything was lowered even further, right? But this doesn't happen with the FMD, meaning if somebody had a C-reactive, if somebody had normal blood glucose, nothing happened. But if somebody was pre-diabetic, now you have a fairly major drop in the fasting glucose. C-reactive protein, somebody started normal, nothing happens, but if somebody's starting with CRP above 1, then you see the effect. Cholesterol below 200 at baseline, nothing much happens, and now big effect. Same thing for systolic blood pressure, below 120 at baseline, over 120. So these differential effects between people that are already doing good versus people that are not doing so good. So people always say then, well, you should compare it to something that's like standard, right? Mediterranean diet. Can you run this 4 cycle of the FMD against the Mediterranean diet? So we did that, and we did this in collaboration with Mark Houston in Tennessee. So we took people from Tennessee that were overweight and that had hypertension, and then we exposed them to 4 months of the Mediterranean diet or 4 5-day cycles of the FMD, right? So a total of 20 days of fasting mimicking diet. And so in both of them, and I don't have time to show it to you, both of them had tremendous effects on glycemia, A1C, and lots of other things, blood pressure, et cetera. But the Mediterranean diet, even after 4 months, 3 pounds of lean mass loss, right? Muscle loss. And this is moving forward by itself. So it's, after 4 cycles of the FMD, no lean body mass loss. And then after 3-month follow-up, so 7 months from the start, now the Mediterranean group had lost 5 pounds of lean mass, which is a lot, right? So in 7 months, losing 5 pounds of muscle, mostly muscle, and some bone maybe, it's a lot. And we don't see much of a change in the FMD, right? So this is addressing what I said earlier. I mean, you have to have differential effects. You want to get rid of the bad stuff, but you don't want to get rid of lean body mass, as it happens, for example, with the calorie restriction that I showed earlier. Okay, A1C, in this trial, we also start seeing the difference between normal people, pre-diabetes, and diabetes. Right? Now we're starting to get in the range of GLP-1 type of drugs. And this was then confirmed by this trial and another trial, one in Germany and one in Holland, that looked at fasting mimicking diet done once a month for either 6 months, like in this trial, or 12 months. And they again compared it to, so they did diabetic patients, and they did a cycle a month for 6 months of the fasting mimicking diet for 5 days, or a cycle a month of a Mediterranean diet. I think they were thinking, fasting mimicking diet, what is that going to do? Let's just try a healthy diet. They're probably going to work as well, right? And sure enough, the Mediterranean diet did absolutely nothing. But the fasting mimicking diet now, A1C drops tremendously over 1 point, and insulin resistance now becomes much lower, right? So they're becoming much healthier. But the most important thing, see, in the Mediterranean diet group, some patients after 6 months increase drug use, and most of them do nothing, and some decrease. So the population is pretty steady with drug use. But in the FMD group, 70% of the patients now drop their drug use. So they're getting the effect, and yet they're reducing the drug, right? And that's what you want. You want to act on aging. You want the body to be more functional, and the diabetes patient should not be stuck managing diabetes, should have the opportunity, not everybody's going to reach it, but have the opportunity to go back to a healthy state. And see here now, we see that it's not just working for diabetes, it's also working for hypertension medication. So Mediterranean diet, you see much more people increasing drug use than decreasing drug use. And here you see the opposite, right? So they're becoming healthier, and that's what we want. Okay, so then what about becoming younger, right? Then can you become younger? So now I would say that the most exciting part of the aging research is this cellular reprogramming, right? Is it possible to turn on what's called Yamanaka factors? These Yamanaka factors basically can take any cell and make it into an embryonic-like stem cell. So make it young and make it able to generate any cell of the body that it wants to. So it's a little bit of science fiction, but very interesting, right? So very interesting. So, of course, if you just randomly increase these Yamanaka factors in cell, you get some benefits and a lot of problems. Why? Because the body is not supposed to all of a sudden, if you're 50-year-old or 70-year-old, you're not supposed to turn on Yamanaka factor. The body doesn't know what to do with it, right? So it just makes a lot of good and a lot of bad. But so we were doing a study on diabetes, and this is diabetes driven by genetic mutations. These mice are DB mice. They're condemned genetically to become very big and to have diabetes, right? And so we start using the fasting-making diet. So they become obese, then they become hyperglycemic, and then they start dying of hyperglycemia, okay, as you will expect. So if we start the fasting-making diet cycles, you see that instead of glucose levels go up, glucose levels come down, and they don't die anymore, okay? So, but this is expected. I mean, not easy to achieve, but very happy about this, but expected. What was not expected is this. So if we do a type 1 diabetes model where, in type 2 diabetes, the insulin is not working very well. In type 1 diabetes, the insulin is not produced. And so we basically got rid of these red cells are the insulin-produced beta cells of the pancreas, right? If you see red, it means they're making insulin, normal insulin. Then we treat it with this STZ drug, and you see now you lose most of the red, and 50 days later here, you have no red, right? So they're not making insulin anymore. So at this point, we start the fasting-making diet cycles. And what happens, you now go back to this, right? So the body knows how to remake, even though this problem is permanent here. They never recover from this. They're permanent type 1 diabetic. But if you start the FMD cycles, now you go back to this, right? And they know exactly what to do, and they go back to making normal levels of insulin. And so how is that possible? Well, it turns out that they turn on Yamanaka factors, right? So this fasting shrinks the system a little bit, and at the end of the fasting mimicking diet period, now these reprogramming factors are turned on, like OCT4, NANOG, and lots of other ones, right? So now the pancreas is getting ready to rebuild itself, right? And then in the refeeding, it waits until food comes back. And then when food comes back, it starts rebuilding. And so you see this green is if mice eat normally, none of these are, green means very low, right? So none of them are activated. And during fasting, FMD, that all of them are activated, right? So it's just a complete program that has the job of rebuilding the pancreas, right? So now imagine if this was true for all organs, that the body already knows how to regenerate itself. And this is suggested by a paper that we just published a few months ago in collaboration with Laura Perrin at CHLA, Children's Hospital Los Angeles. So here's a whole kidney, a slice of the kidney, right? You see that it's got a specific structure, and you can see it here, and this is called spatial transcriptomic. So now we treat it same as we did for the pancreas. We treat it with a toxin that you see disrupts this architecture, right? Completely disrupted, and you see it here too, right? From here to here, completely disrupted. Then we start the fasting-making diet cycles, and you see what happens. After 6 cycles, it goes back to normal, right? So again, the kidney, it knows how to, it already has all the information to go back to its normal state, even though you start from complete chaos here in the middle, and as you can see from transcriptomic analysis. So now we're like 6 or 7 different organs and systems. We see the same, right? So it means that probably everywhere, right? So, and look at what happens to patients, right? So now these are kidney disease patients, and they, well, first of all, CRP is reduced in the patient, but look at proteinuria. It's one of the markers of non-functioning kidneys, and now it drops from above 2 to a little bit above 1 on average. But more importantly, 1 year after, and this patient only did 3 cycles of the fasting-making diet, and a year later, the kidney is still functioning better, right? Just by doing 3 cycles of the FMD. Now, it's a small study, but we had 2 arms that did the FMD at 2 different times and got identical results, suggesting that. So now we want to do a much larger study on kidney disease patients. Okay, so then what about aging itself? So we in collaboration with Morgan Levine, who's now at Altos Lab in San Diego, we did this study looking at what is the biological age. Can we actually reduce the biological age of people after 3 cycles of the fasting-making diet? And sure enough, just 3 cycles of the FMD are sufficient to reduce biological age by 2.5 years, right? So this is within 2 months, you're getting effects. I mean, she measures biological age by this method It's called BioAge that she developed at Yale. And so based on that method, now you have a reduction of 2.5 years. So people are becoming younger, and this is from 2 different trials, identical results from 2 different clinical trials, almost 200 patients. So, so a lot of what I showed you that probably has to do with making systems younger. Okay, so then quickly I'll talk about cancer. And I just published a book on cancer called Fasting Cancer. And so I wanted to say a few words about cancer. And so this is a lung cancer model and a breast cancer model. This is actually not our work, and this is our work. And so, and this is looking at cancer-free survival, right? So a lot of research is based on, can you slow down the cancer growth? Here we're talking about, can you cure the mice permanently? So if you give them chemo or fasting, both the lung cancer and the breast cancer model, you see no or very few mice that become cured, right? But look at what happened when you combine the fasting cycles with the chemo. So now you have 60% or so of the mice becoming cancer-free, right? So the combination of the two are now able to search and destroy every cancer cell, no matter where they are. And so now this will scare you, but this is what we call RNA-seq, right? So forget about the detail. The point is now we can use technology to identify what we call starvation escape pathway. So what I showed you earlier is like old style, right? Chemo plus fasting, and it's working very well. But the new technology is like, well, let me find out how the cancer cell rewires itself in response to fasting, and then target the pathways that I know are important for that cancer cell. This can go very fast, right? Within 1 day, we could tell you how any cancer is being rewired. And so Julia did this in the lab. I have a second lab, one at USC, one in Milan. And you see this is cancer. This is triple-negative breast cancer growing very fast. And then we use that technology. We identify the pathway. We target it with 3 available drugs. And now the cancer comes down and stays down for a very long period, right? So we can actually reverse the course of cancer progression. And so this is the clinical trial, and Vernieris just published a paper that confirmed these earlier results. These are women that have triple-negative breast cancer, the same one I just showed you. And so the survival after nearly 4 years is almost doubled in those that do fasting-making diets plus chemo versus chemotherapy alone, right? So the initial data and the new data that he just published just a month ago now is suggesting that it works. It works in patients. It can make a big difference. You know, let's see if we get the cancer-free survival that we see in the mice. But certainly, you know, having a doubled survival at 4 years is already a big deal. Okay, and then another thing that was interesting, another trial that they ran, the oncologists at the beginning were very skeptical when I first talked to them, you know, over 10 years ago, or 15 years ago, actually, about 15 years ago. They were very skeptical. So the same oncologist at the National Cancer Institute in Milan eventually did lots of trials on this. And in the first trial they did, out of 101 patients, there were, I think, 20 metastatic, stage 4 metastatic cancers. And they were very surprised that 5 out of the 20 went into a complete and long-lasting tumor remission, right? So they were so surprised by this, they ended up publishing the paper saying, you know, colorectal cancer stage 4, breast cancer, even pancreatic cancer stage 4. All of a sudden, in lung cancer, these patients went into remission. I mean, you know, you don't expect, this can happen from time to time, but 5 in the same trial, it's very unusual, right? And this is why they published this exceptional tumor responses paper. Okay, so then what about everyday diet? And so we talked about proteins, right? So proteins are driving this growth hormone, IGF-1, TOR, that are increasing the levels of these pro-aging pathways. And so we did a study years ago using the the CDC database, the NHANES is called, and I don't know why this is going forward. And so the people in the US that report eating less than 10% of calories from proteins had there was a 4-fold reduction in the risk of developing cancer, right? And a 75% reduction, I mean, a 75% increase in those having a high protein diet, increase in overall mortality, a risk of overall mortality. But when we look at the 70, 80-year-olds who reported, I have a very low protein diet, the opposite happened, right? They did worse. So old people that were eating a very low protein diet did not do well at all. In fact, did worse than those that had a moderate protein diet, right? So we started to think about, this is probably age specific. And this is why at 65 or so, you start seeing in the US population a decreasing weight, right? On average, Americans start losing weight at age 65, right? And this is probably when they enter this high frailty state. And in the high frailty state, now the low protein diet is now turns from good to bad, right? Okay, so I'm showing this lady from Okinawa. She was 90 years old. She was dancing 3 times a week. But the point is that Okinawans for the longest time had the record longevity in the world, and or record life expectancy in the world. And not surprisingly, their historical data was extremely low on protein. They ate about 36 grams of proteins per day, right? Less than 10% of their calories came from protein. And so not surprising, right? But probably frail, just like the Italians, right? So this is Salvatore Caruso. He was 108, and he was still singing songs by memory. So he was very healthy. But same thing in Italy, mostly vegan, high carb, low protein diet, right? So they gave him longevity, record longevity, but record frailty. So the Southern Europeans are the most frail of Europe, right? So they live long, but they're frail. They're weaker, right? So that's of course what you want to fight. And so you want both, right? You want to live long, strong. You don't want to live long, frail. And so probably the Okinawans had the same problem because they had a very low calorie, more like the monkeys, right? They were calorie restricted, low proteins, right? So extremes that probably are not good in modern times. Okay, so then what is the longevity diet? Well, it's mostly plant-based, say vegan plus fish 3-4 times a week, but low protein until age 65, 70, and then moderate protein after. So you have to eat more and more variety. So now the white meat may become a good option after age 65. Legumes, whole grains, vegetables, olive oil, nuts, dark chocolate, salmon, no red processed meat, low eggs, low sugar, low refined grains. This is a controversial thing, but it turns out, so you hear a lot of in the news now, oh, stop drinking alcohol. But it turns out that alcohol is actually neutral for lifespan, right? So it's got a neutral effect over and over and over in all the studies done. Some of them are positive, some of them are a little bit negative, but overall, 3 or 4 glasses a week are neutral, right? If you go above that, then you start seeing a consistently negative effect. But 3 or 4 glasses a week have a neutral association with lifespan and life expectancy. And so with the exception of people that have risk factor for certain cancers like breast cancer, right? If you have already a risk factor for breast cancer, alcohol does increase the risk of slightly the risk of breast cancer. You don't want to combine 2 risk factors, right? If you already have in the family a high rate of breast cancer, and there's 3 or 4 cancers, very easy, you can Google it, which are the cancers that are affected by alcohol. I think esophageal and a few others. If you have risk for that, that's probably not a good idea to drink at all. So high nourishment, omega-3, vitamins, minerals. And then, for those of you that listen to Sachin Panda, 12 hours probably is a good compromise. 12 hours of eating and 12 hours of fasting. So if you start at 8 a.m., you have to be done eating by 8 p.m. Okay, so it's very simple. Most people, centenarians, always done this, right? So the 12 hours. So this is very safe. It's not just very effective. Not as effective as if you did 16 hours, but much safer and much easier to do. And so, so this is something I've been doing for a long, long time. So Monday through Friday, I have a healthy snack instead of lunch. And this is the way I keep a normal weight. Otherwise, I will have gained weight like everybody else. And then periodic medically supervised. It doesn't have to be necessarily medically supervised. if the person does not have any significant medical problems, but best if medically or dietitian supervised fasting mimicking diets that I just talked about. Now, this just came out, even though, of course, I've been talking about the longevity diet for many years, but this came out of Norway, and they looked at lots of the ingredients that I just told you about, legumes, whole grain, nuts, right? And either having positive effects or negative effects on lifespan. And so now, something very similar to the longevity diet, they show it's associated with 11 to 13 years life expectancy increase if started at age 20, and 8 to 9 years life expectancy increase if started at age 60, and still 3 to 4 years life expectancy increase if started at age 80, right? So it keeps working less and less as you move forward, but still 8 to 9 years at age 60. So it's an impressive, so obviously it's not just living 9 years longer, it's about also living much healthier in that period, right? So if somebody started at 55, you may look at 30 years of being much healthier and maybe an extra 10 years of life, right? So it's not bad. Okay, so this is just a summary. So this is showing chronological age versus biological age. So lots of factors can increase your biological age and disease risk. But we now are starting to get more and more evidence that the longevity diet that I just discussed, and the cycle of the fasting mimicking diet, but also the time-restricted eating that Sachin Panda is talking about. I really like those three, right? And keep in mind that longevity diet has to be personalized, right? Meaning that somebody from Japan is not going to have the same longevity diet. This is why I don't like the idea of Mediterranean diet. Mediterranean diet is for people from the Mediterranean. So different people from different places are going to need their own version of that. And that's very, very important, because I always say somebody can eat tomatoes all day long, and then a person next to them eats some tomatoes, and they have a terrible reaction to tomatoes, right? So this is why it has to be personalized. And of course, I didn't talk about exercise, but 150 minutes of exercise plus training, muscle training as regularly as possible. And that's my recipes for longevity extension. Thank you.